What would happen if there was a mutation in a transfer RNA such that it allowed it to bind to a stop codon? Here is such a mutation. The anticodon goes from AUG as an anticodon to AUC as an anticodon. So now it will bind U, A, and G as a codon. Well, you know UAG is a stop codon, and so now we have a mutant transfer RNA binding to a stop codon, so this translation will no longer stop. It will produce a different protein, therefore a different protein will have a different function. This is an electron micrograph of something called a polysome. This line just down the middle here, that is the DNA, that's a gene, and then there's lines coming off here, that is messenger RNA, and then attached to that messenger RNA are ribosomes. The point of this diagram is to show that you can have multiple RNA polymerases, depicted here in red, and multiple ribosomes, all happening at the same time. And so in this particular case we have simultaneous transcription and translation. Does this happen in eukaryotes, prokaryotes, or both? That's the question. An additional component of making a protein, here is a ribosome. These are ribosomes right here. And this is messenger RNA, and here are the protein chains being made. Proteins can be moved into particular areas of the cell, and in this case this protein is being moved into the endoplasmic reticulum, and it knows to go there because it has a signal sequence. This signal sequence is the first few amino acids on the amino terminus of the protein, and it's a bit like a zip code. It directs the protein to go to a particular place in the cell. Francis Crick had a lot to do with discovering DNA and other molecular processes, including something called wobble. Wobble is where a transfer RNA anticodon, you can see here AGG, it may bind to codons that are not exactly correct. In other words, you expect the A to bind to a U and a G to bind to a C. And you expect the G to bind to the C, but you would not expect it to bind to the U, but it does. And so wobble is where an anticodon can bind not only U, C, C in this case, but also U, C, U. It is where the 5' anticodon nucleotide does not have 100% fidelity with the 3' codon nucleotide. If we look at this diagram here, we can see that the anticodon position there, G, will bind to U or C, as we just saw, and if it's U, it will bind to A or G, and if it's another nucleotide, this is a 6th nucleotide, it's called inosine. It's an I. Inosine will bind to U, C, or A. So we have successfully transcribed a DNA gene and successfully translated that messenger RNA product into proteins. Once these polypeptides fold into a three-dimensional structure, as we mentioned, they have a particular function. And so the difference between a polypeptide, which is a chain of amino acids, and a protein is really function. If you have a mutation whereby that folding is prevented or changed, then that function will be changed, and that could be the basis of a disease. So therefore, again, if we change the DNA, we change the RNA, we change the protein, and we change your phenotype. So maybe changing a protein is a good way to kill something, right? And so a lot of, well, these three different antibiotics, tetracycline, streptomycin, and erythromycin, if we add those antibiotics, they affect transcription or anticodon interaction or ribosome movement in bacteria. Why don't these antibiotics affect humans? So protein structure and protein folding is a very important component. And you should remember from your freshman days that there are 4 levels of protein structure. The primary structure is the sequence of amino acids in a polypeptide chain. So depending on which amino acids they are, here's 1, 2, 3, 4, and 5. If they're all different, that gives you one particular primary structure to a protein. If, on the other hand, we make this number 7, so it goes amino acid 1, 7, 3, 4, 5, then we have changed the sequence of amino acids. You change the primary structure. The secondary structure is what shape this primary structure folds up into. And if we look at this diagram here, here's the primary structure sequence of amino acids, and we can fold up this sequence into a helix, and we call this an alpha helix, or we can fold it up into a sheet where it just folds back on itself, and this is called a beta sheet. There. The similarity between these two is that hydrogen bonds hold them all together. Hydrogen bonds are these dotted lines holding the beta sheet and the alpha helix together. The tertiary structure is by taking these sheets and helices and folding them up upon themselves to produce an even more complicated structure. And some of these components could be disulfide bonds, which are quite strong bonds. They could also be hydrophobic interactions holding these components together as well. Finally, the quaternary structure is where you have two or more polypeptide chains interacting together. So you've got separate interaction of these polypeptide chains. And here it's hemoglobin, and in this case we have 4 separate polypeptides, all shown here in slightly different colors. 3 and 4 down there. Those 4 components, 4 separate polypeptide chains coming together to make a protein with quaternary structure. Quaternary does not mean you need 4 polypeptide chains, it just means there's a 4th level of folding. So what happens if we do change some DNA? And here is the classical example. This is a DNA sequence here, GAG, the opposite strand is CTC. If we mutate that so that the middle base is changed, instead of being an A here, it's a T, then the RNA will be different. Being GAG, it now becomes GUG, and the protein is different because this amino acid goes from a glutamate to a valine. And if you change the primary structure of a protein, it changes the shape. And so the shape of the hemoglobin protein, in this case, goes from being globular to being clumped, and here. And this clumped hemoglobin gives rise to a sickle shape to a red blood cell. And that will be this one right here. This one is normal, and this one is sickle. And of course, if this happens, you have sickle cell anemia. And it's due to a single base change giving a single amino acid change, giving a change in shape and therefore function to the resulting protein. Another example of a protein disease is when some proteins can fold in a different shape. And there are 2 ways of folding. In this particular case, there's an alpha and a beta way of folding, and that's in these diseases called prion diseases. A prion protein can fold here. You can see these 2 different shapes here. And prions are proteins that are normally in your cells, and in some cases, if they fold in the wrong configuration, they can destroy that cell. And this is a photograph of a slice of brain tissue, and you can see the holes in the brain tissue here. This gives a spongiform-like appearance to brain tissue, and so it's called spongiform encephalitis. If it occurs in cows, it's called bovine spongiform encephalitis, in other words BSE or mad cow disease. And that completes chapter 9.